Okay, so there's this three scenarios where my cells are present. So you have the cell that again depicted at 300 milliosmolarity, which is our ideal environment for a cell. Red blood cell under normal condition is 300 milliosmolarity. And what I'm changing is the solution, osmolarity of the solution at 250, 300, and 450. So again, I'm going to use first the word osmolarity to define both my cell and solution. I compare the two numbers. In this case, my cell has the larger number, so my cell is hyperosmotic, and my solution is hypoosmotic. Now, in terms of tonicity, again, I'm only focused on the solution. So solution has the smaller number, so my solution is hypotonic. Now, the next part of this concept comes into, again, which direction your water is going to move. And for that, we're going to rely on the concept of osmotic pressure. So wherever you have more solutes, the water is always going to move in that direction, guys. Wherever you have more solutes, water is going to move in that direction. And again, the way I always think about it, and this was also presented in your lecture, is if you eat something sweet or salty, which means you're taking a lot of solute into your system, almost immediately your body desires water. So water is required or follows the solute. Wherever the solute goes, the water goes in that same direction. If I have more solute in one place, water will follow in the same direction. So look at this scenario. My cell has the larger concentration of solutes, so the water is going to rush into my cell. And if there is so much water that going into the cell, first the cell starts to basically get bigger or bloated. And if the amount of water is significant, then that's going to cause the cell to basically burst. So in the microscope, when you look at it, your cell is again either bloated or kind of more circular. And if they have basically been destroyed or burst, they are not visible, or they appear as what we call ghost cells. And I show you a picture of what that looks like in just a second. Now, the second scenario, everything is equal, right? Your cell osmolarity is equal to your solution osmolarity. So in this scenario, every word that I'm going to use is iso. So it's isoosmotic cell, isoosmotic solution, and isotonic solution. Now, in this scenario, again, water is going to move in and out of the cell. The difference is that the amount of water that gets in and out of the cell is equal. So the net, there is literally a net zero movement of water. Consequently, your cell shape will not change. Basically, it stays the same because there's no net movement. Last scenario, you have a larger number of particles inside your solution. So your cell has a smaller number, so it's hypoosmotic. Your solution is hyperosmotic because it has a larger number. And again, because it's larger number in a solution, it's also called a hypertonic solution. Now, where you have more solute, water is going to move in that direction. So in this scenario, again, the water gets pulled out of the cell, and that causes the cell to basically crenate or shrink. The way your cells look like under the microscope, they kind of look like little spiky cells. Their size is smaller, and they have this little like pointy structures sticking out of them. So here, exactly what I'm describing for you here. Hypotonic, you barely see any cells, and what you see are very light. So this is what I call a ghost cell. What it used to be a cell, but it's already burst, and you just kind of see the outline of where that cell used to be. Isotonic, my cells look beautiful and round and gorgeous. There's no difference in terms of shape. And then last one is where the water has left your cell, and now your cell has basically crenated or shrank, and you have these little spikes on it, and the size are smaller. So when you guys are doing your experiment, the well, you're answering question about your experiment, you will get basically the same concept. So one thing that you need to remember when you're answering those questions are the following. What we consider normal for our red blood cells when you answer these questions is 300 milliosmolarity for the osmolarity of the red blood cells. And that's equal to having about 0.89 or 0.9% sodium chloride, which basically means salt. So when you're predicting what is happening to your cell based on the solution, if the solution that you placed in has anything below 0.9% NaCl, that's considered a hypotonic solution. If it has 0.89 or 0.9, it's isotonic solution. And if you have anything that has more than 0.9% NaCl in it, that's considered a hypertonic solution. So again, in the first scenario, you're gonna see typically bloating of the cells or ghost cells. Second scenario, will not expect to see any change. And third scenario, you expect to see your cells shrinking or crenating as a result. FYI, in an exam, guys, this is a really common mistake I see among the students. If you have either 0.89 or 0.9%, for our purposes, those two numbers are the same, and they're identified as isotonic. So if I give you 0.89 or 0.9%, assume they're the same, and again, they're isotonic. Any question or clarification on anything we discussed so far? You're welcome to unmute yourself and ask the question, guys. So let's move on to our other experiment. Thank you. Our other experiment is osmotic pressure, which I already mentioned about when we were talking about the earlier experiment. But basically, osmotic pressure, again, it means when you have solutes and those solutes are going to pull water in that direction. And again, the more solutes you have, the more desire you have for that water. So eating something sweet or salty makes you want to drink water. And the more chips or sweets you eat, the more water your body desires. So it's a direct relationship again between the two. So when you're looking at our experiment, one of the experiments you guys are going to do today is looking at 3 scenarios with 3 different solutions of sugar. One is 10% sugar, the other one's 20%, and the last one is 30%. So what you're doing basically, if you were to do the experiment, is you would use a dialysis bag which simulates a cell. So again, we can use an actual cell. So we use something that simulates the cell. And basically this dialysis bag have these little tiny holes that allows the water and the small molecules to get in and out. So I will basically pack my little baggies with either 10, 20, or 30% sugar solution. I tie the ends, and then I weight the bag individually. So it's really important to have that initial weight because we want to see how much water actually moves in and causes the baggie to gain weight. Now you're going to put each of these baggies at the same time in the basically pure water solution and leave them in there for about 30 to 45 minutes. After you're done, take the baggies out and you basically put them and measure their weight for each of the bags. Now, the idea is I don't really care what was the initial weight and what was the final weight individually. What I care about is percentage change, which simply means I want to know how much percentage-wise they have gained weight. And I wrote the equation for you here. It's also part of your lab, so you guys can take a look at it. But basically, you take the initial weight, subtract it from the final weight, and divide it by initial times 100. Now, at the end, when you finish this, you're going to graph only the percentage change. This should not be a new concept. Basically, you are going to use a bar graph, and you're going to have 3 bars on that graph, one for 10%, one for 20%, and one for 30%. And you're representing only the percentage change, guys. Only and only the percentage change would be depicted here. Now, the idea here is, again, I'm just gonna put your mind into the right track. As my percentage of sugar increases, as there is more solute present in my environment, the osmotic pressure would be stronger, and therefore more water is gonna move in that direction, causing the greatest percentage change. So in this scenario, I will have the 10% having a difference, having a percentage change, but has the smallest percentage change. The 20% is going to have a higher percentage change, and the 30% is going to have the greatest percentage change among the three solutions. Does that make sense? No questions, so I guess I'm good. Now, the last thing that I want to talk about, and this was an actual instructor demo if you guys were in class, but I still want to go over it. There is exam questions on basically every part that I've discussed with you guys so far. So make sure you guys spend time on this lab. It's really important. So when we're talking about membrane permeability, which is literally the first thing I talked to you guys today, one of the factors that affect the permeability of your membrane, how things get in and out of your cell, is based on the size of the molecules or size of your solute particles. So the smaller molecule, the one that are typically have no charge on them, they can easily get in and out of your plasma membrane and basically balance themselves. Versus when we are looking at larger molecules like particles that are charged, like a starch, they basically remain where they are because of their large size or the large molecular weight that they have. Or large, not molecular weight, sorry, the large size that they have. So in this experiment, we are basically going to look at a starch and iodine and water and see, based on their movement, are they considered to be large or small particles? So for this experiment, we're using a starch and iodine because if you guys recall from our second experiment this semester, we have used iodine or Lugol iodine as our indicator to identify the presence of a starch. And we said iodine normally is yellow in color, and when it interacts with the starch, it turns to that bluish black. So we're going to use that feature to see if molecules actually move in and out of our environment. So basically what I'm going to do is I'm going to take a bag of one of those synthetic cells that I mentioned earlier. I'm going to fill it up with a starch solution. And if you guys have ever seen a starch solution, it has that kind of white color, almost cloudy looking solution. And I'm going to weight my little baggie. And I'm going to place that bag inside a container that is filled with pure water and a few drops of iodine. Again, because this is just water and iodine in my environment, I'm starting with a yellow color solution. So at this point, my initial weight is, this is arbitrary number, by the way, don't use these, use that one that I'm actually giving you in an experiment. I'm starting about 3 grams for the starch bag. The initial color of the starch bag is white and cloudy, and the solution color is yellow. Now, again, I'm going to leave this sit for about 30 to 45 minutes, and then basically record the final data. Now, a few things that you will identify here is first, is the final weight of the starch bag has increased. So we went from about 3 grams to 4 grams. The second change you notice is that the starch bag, which was originally just white color, now has turned black. The last thing is that the solution color, which was yellow, is still yellow. So we have 3 components that we're testing: water, starch, and iodine. And we have 3 data points that we basically can use to assess that. So let's start with the easiest one. The solution color was yellow and now it's still yellow. What does that mean? It basically means that the starch was not able to get out of the bag to interact with iodine to cause any color change. So that basically means a starch was large enough that couldn't pass through the membrane to interact with iodine. Time to type and talk at the same time. Interact with iodine. Therefore, the solution color remain yellow. Now, the second thing, again, we're going to observe is the change in weight, right? So that's another thing that changed. So change in weight literally implies that the water has moved in. There's a significant water moving in. So change in weight basically indicate water movement into the bag. And again, since we are talking about water, the means of water movement is always going to be osmosis. It's diffusion, but it's specifically osmosis. And that causes the change in weight. And again, what does that tell you about water molecules? Size are small, right? Because they were able to basically get into your starch solution and cause that baggie to gain weight. The third component, iodine. Iodine was able to also get into the starch bag because we went from cloudy to black. So iodine got into the bag, which basically means iodine molecules are small, right? So they were able to get into the baggie and cause that color change to happen. Now, one of the common questions I usually get at this stage when I was in class is, why can't I say that the weight of the bag that changed from 3 grams to 4 grams is not the result of iodine getting into the bag? Well, isn't it iodine getting into the bag? Why can't I associate it with the weight gain in the bag? The main reason is this, guys. If you recall, when I started this solution, I said I fill this container with water and only a few drops of iodine. So few drops of iodine is so insignificant in terms of weight that it will not have any effect even if it gets into the starch baggie. So we are using like such precise instrument that measures the smallest amount of change. We're kind of looking at it as a big picture here. So the change in weight can only be associated with water because that's the only other molecule we have outside the environment. And iodine change or iodine movement can be measured by the color change rather than weight gain. Questions about this, guys? This is the last thing I want to talk about with this lab before we move on. And I'll walk you guys through the actual experiment. Okay. I don't hear any questions, so let's go ahead and I'm going to walk you guys through the actual assignment. Let me just share my screen with you guys again. Okay, so this is your experiment for today. Again, most of the thing I just discussed with you is part of the introduction. Please do review it for your exam. You may have questions specifically to the introduction section. There is a video for diffusion. I really like this video, so it's really informative and really fun to watch, actually. You have this video, which is cell membrane transport. This is the video that you're actually going to watch to get data for the experiments in your lab activity today. So watch this video specifically. The PowerPoint I was just showing you here is this PowerPoint that says membrane transport. And then for today, you're going to do the lab activity. So first part is basically you looking at osmolarity versus tonicity. looking to see how each of these solutions actually affect your cell. Again, the whole concept of hypertonic, hypotonic, isotonic, and then what is the appearance of those cells under the microscope. This experiment is based on how diffusion rate is affected based on molecular weight. Again, search for molecular weight of these two components by using a Google search. So all you need to do is say methylene blue, molecular weight, whatever number it gives you, type it right here. Color change for the starch bag is the last experiment I just finished talking about with the whole iodine going into the bag. Again, collect the data, initial and final, from the video that I pointed out earlier. And last one is the 3 sugar solution. Again, initial and final weight are going to be available in your video presentation. And then percentage change is something you calculate. And when it comes to graphing, guys, again, when you graph this, I'm giving you the option to either use an Excel if you feel comfortable using the Excel to graph the percentage change with only 3 bars on it. Or you may just do a hand-drawn graph and then basically attach it as part of your lab. Either one is fine with me as far as the data presentation is correct. That's my goal. Like my idea is not for you to draw this beautiful graph on Excel and it's all wrong. Rather, it's did you understand the concept and understand what you're actually drawing and interpreting your graph data. Okay, this is it for my part. If there is any questions, the floor is open. Otherwise, it will take me a few minutes to put you guys into your group because I didn't get a chance to do it beforehand. And then we'll open up your breakout rooms to work with your group. No questions?
Good morning, Professor. This is Lily.
Good morning, Lily.
I have a question. Where can I find the recorder